back to Medical Point. This is our second lesson. In the first lesson, we discussed about whether or not viruses are alive. This week, we will be covering the viral replication cycles. And again, feel free to pause the video or rewatch if you need to. Before jumping into the lesson, let's do a quick overview. Viruses are made of only proteins and one type of nucleic acid, which means they have no organelles or any machinery to make copies of themselves with. So they have to get inside other cells in order to use their energy, or known as ATP, and their organelles to make copies of themselves. Just for those who are wondering, while discussing this topic with others, some big scientific terms may be used. So here are just some key vocab that may or may not be used in this video. Feel free to pause if you need it. Moving on, to begin with, how does a virus get into the cell? There are 3 ways a virus can enter the host cell. The first way is through bacterial phages, which is them injecting their viral genome into the host cell. Viral genome is the virus's genetic material. It is diverse, meaning it could either be DNA or RNA, but not both. The second and third way for viruses with and without envelopes. Envelopes are the outer layer of a virus that helps the virus enter the host cell. It's kind of similar to a cell's membrane. The second way is for non-enveloped viruses. They trick the cell's receptors into letting them in. The third way is for enveloped viruses, in which they directly fuse with the host cell's membrane. Using one of these 3 ways, the virus gets inside the host cell and is ready to take over through the lytic or lysogenic cycle. First, we will talk about the lytic cycle. In the last lesson, we gave a brief summary of how the lytic cycle works, but this time we will give a more in-depth description. The lytic cycle is for viruses who want to start replicating at the get-go. In the lytic cycle, the virus takes over the cell's machinery, such as ribosomes, nucleic acid, amino acids, and many other things. The virus takes over such components to start making copies of the virus's genetic material, which is either RNA or DNA. And also, the virus starts making copies of the virus's protein in order to make a protein coat. Then the virus makes more and more copies, which can cause the cell to break open, or in other terms, lyse. After it lyses, all of the viruses get inside the host cell, release into the environment, and start to affect nearby cells and slowly take over the organism. Once this process is complete, the host cell will die. Dun dun dun. Next, we will cover the Lysogenic cycle. Last time we mentioned that the virus can switch between the lytic and lysogenic cycle in order to adapt to the host cell's condition. This means that the virus that goes through the lysogenic cycle will take a break and let the cell live longer. Viruses can carry out the lysogenic cycle in eukaryotes and prokaryotes. In order to sneak into the host cell without it noticing, the virus will inject its DNA into the host cell. The viral DNA will combine with the DNA of the host, so the virus can't tell it's there. The virus needs the host to replicate its DNA since it has repressor genes. That means that their DNA cannot be expressed or transcribed. This is also known as the provirus, as it isn't doing anything, and because of that, this phase where the virus just waits is called the dormant or latent phase. The host cell continues to carry out normal cell functions such as DNA replication without realizing the virus is actually there. This way, the viral DNA will replicate each time the host cell replicates. In prokaryotes, the host cell reproduces asexually, so the virus DNA will be in every new organism that is produced. This also helps the virus spread. Anyways, the virus continues replicating in both the eukaryotes and prokaryotes. Unlike the lytic cycle, the lysogenic cycle does not kill the host. Instead, it tries to keep the body alive so the virus can live inside it. Ever so often, if the host cell might get exposed to UV light occurs or or another like bad condition, this will cause the repressor genes to weaken, causing the process to change back to lytic. Then it will produce the capsids for all the viral DNA it created, and then the host cell will lyse and die. Poor host cell. I hope this lesson was also helpful to study for the MCAT or just for any regular science class you might have. Make sure to subscribe to the Medical Point YouTube channel, and thank you so much for watching.